Did you turn it on?
I turned it on, thank you, Kate. Welcome everyone, it's nice to see you all to the latest edition of the CCOVI lecture series. It's my pleasure to introduce Wes Pearson today. And I enjoy the irony, a New Zealander resident in Canada introducing a Canadian resident in Australia. That's the nature of international research these days. Wes is a senior sensory scientist working at the Australian Wine Research Institute in Adelaide, Australia. He is a Canuck from Winnipeg originally, and he's been part of the sensory team at the AWRI since 2011, and I think in Australia since 2008. He has a BSc in Wine Biochem from the University of British Columbia and a diploma in Consumer and Sensory Science from Davis in California. Prior to joining the AWRI, Wes has been a travelling winemaker working all over the world, including Okanagan Valley and Bordeaux. And when he's not conducting sensory research, he's also the proprietor and winemaker at a McLaren Vale winery called Dodgy Brothers. Fancy an Australian winery being called Dodgy. His talk today is on green flavours in Cabernet Sauvignon wine, and I think Wes will also give a bit of an introduction and overview to the AWRI. So welcome, Wes, and we look forward to your talk.
Thanks for having me, guys. Yeah, like Gary said, I'll start by talking a little bit about the AWRI and what it does and why it's cool and why it's different from a lot of research organizations around the world. The biggest thing about the AWRI is that I would say that it's owned by the Australian wine industry. And so what that means is that most of our funding that comes for all of our research comes from a grape levy. So if you process fruit, if you buy or make wine from grapes in Australia, for every ton of grapes that you process, you pay a levy to the government. The government collects that money and then distributes it as they see fit for R&D, marketing, what have you. The AWRI collects part of that levy to fund a lot of our research. We also have a commercial division. We also have the largest wine lab in Australia. So we do have some commercial ventures as well, but for our research, we're basically funded by the industry. So we are not an academic institution. So that gives us, certainly there's, in academia, there's certain issues that we wouldn't have to deal with in regards when it comes to funding our research projects. So, but that also gives us sometimes a different mandate when it comes to the kind of research that we do. The research that we do has to be applicable to industry's needs, meaning we want to see our research get uptake into industry quickly and efficiently. So that's always a mandate when it comes to thinking about how we're going to do our research. How will I explain that? It's the benefit of our research has to be shown in industry. And the people that are making the decisions in the big wineries, unfortunately they're not the winemakers, they're the bean counters. So it's always a struggle to be able to show people, look, this is going to improve your business. So that's part of what we try to do with our sensory group at the AWRI. We're always, we pretty much attach a sensory component to all the research that goes through there. And oftentimes we'll also attach a consumer element to the sensory research that we'll do. So now if looking at that from the perspective of uptake into industry, if you're going to do a research project, could be a million things, whatever it is you choose to do. You do your experiment, you run the chemistry, you have a look at everything. Oh, okay, looks like we succeeded in trying to do what we were going to do. Now we attach a sensory component to that. All right, we're going to check through all the sensory. Yes, we can prove that, yes, in fact, there is a sensory difference. People can see the difference. And now we're going to attach a consumer element to that. We're going to try this whatever this treatment might be. We're going to try this with consumers. And yes, the consumers, they like this product better. So now you've got this piece of research. What we've tried to do works. It shows a sensory difference, and consumers like it better. Now you're going to present this to industry. Now you've got some traction. So this is something that we try to do. This is a model that we try to follow because it's efficient, and like I said, the bean counters say, oh, consumers like that better. Well, all right, that's at the end game. That's going to sell more wine. So that's what we try to do with all our research, and that seems to be an effective model. The Institute basically is 3 groups. We have a research group, which is about 50 staff, covering all kinds of different topics, microbiology, so yeast research. We also house it's 1,000 deep, I believe, the collection of different yeast species from, like Saccharomyces to Brettanomyces species, MLF bacteria, all kinds of stuff. We have a big collection that we keep everything in. We also, part of that research group, we have a flavor chemistry group as well. And that covers, the sensory group does fall into that, I think, but we also have people doing tannin work, color, all kinds of things in that group. Another portion of the Institute is industry development services. So that's kind of our liaison group with the industry. So like I was saying before, we are kind of owned by the industry. If you're a winemaker and you pay a grape levy, that entitles you to any of our published research you can read online. You can use our facilities, our library. And if you have any problems with winemaking, it's actually like a hotline. You can call the winemaker hotline and talk to an enologist and work through your problems. And whether that's sending samples in for sensory analysis, try to figure out what your problem is, or looking at different options for what it might be. It's not a consulting service, but it's a problem-solving service. And that's something that we offer free. If you pay your grape levy, if you processed 1 ton of grapes and paid $9 to the government for your grape levy, you have access to all of these things. So that's something that that group does. And it also does puts on road shows across the country for in all the different wine regions. What research we currently present are we undertaking? What have we finished? What's out in industry? What problems are people seeing? All these things. So usually we do about once a year in every region in Australia. We'll put on a road show, present some different stuff, and get all the winemakers together and have a chat with them, see what problems they're experiencing and what they think about what we're doing and where we can go. So that's kind of how we keep in touch with the industry, or one way that we do anyway. We also run a program called the Advanced Wine Assessment Course. This is a program that we run twice a year, and usually takes about 2 or 3 years to get in once you sign up. And it's about $4,500, and it is the most intensive wine tasting program that you've ever done. It's 4 days straight of tasting about 120 wines a day from all over the world. It's basically how to be a wine judge is what the course is about. So it's for winemakers, wine trades, sommeliers, people like that who are interested in doing wine show judging. And so we put this course on, and it's a lot of fun. It's you can imagine people pay $5,000 to taste wine for a week. So, and we get to do that 2 weeks a year. Basically, we shut everything down and just put this on for these for these wine professionals. So that's quite fun. That's something pretty cool that we do. Sensory group, what I do basically, we have 3 panels that we basically use for most of our day-to-day stuff. You could imagine we would see all kinds of stuff from research, industry problems, also commercial companies that want us to do, look at different things, oak companies looking at different oak treatments, agricultural products. Is this new spray or this new product going to cause some sensory differences? So testing through there. So there's some commercial stuff that we look at. But we have 3 panels that we basically use. The first is our descriptive analysis panel. You would have one here. I'm making that assumption, Gary, but is that the case?
We do a lot of DA, but we don't have a...
You don't have a particular panel? So we have a panel that are employed by the AWRI, and they're not professionals at all. They're just wine tasters, basically. We recruit them from the public. They come in 3 days a week for 2 hours a day and taste wine, basically. That's their job. It's not bad. There's been worse jobs out there. And so we use them as, I don't know if a lot of you are familiar with descriptive analysis, but it's a tool that we can use where we can train up the panelists to be effective tools to measure things. So like you would calibrate a pH meter in your lab, we calibrate the panel to measure something as well. So they measure the sensory attributes for us, and then we apply statistics and tells us a story about what they've been tasting. So we use that panel to do probably the bulk of the work that we do. Most of the research projects that we do, we use the descriptive analysis panel. And we have a second panel, which is it's a professional tasting panel, effectively. Everyone on the panel has either winemaking experience, wine show judging experience, people who have come from industry who work at the AWRI, and they are a technical quality panel. And so what we use this panel for is for mostly industry problems. So if you've got a problem in your winery, you're not sure what it is, you're not sure where to start, you would send your samples into the AWRI. And the first thing that we would do is sensory analysis with the quality panel. And so we would have 15 or 16 judges sit down to taste, smell the wine, and write a detailed note about the wine. Then the oenologist would collect those notes, read them, and from there they would make an assessment on where they might want to go to try to investigate what the problem might be. I mean, you could take that bottle and you could run a whole bunch of analysis on it in the lab, but that's expensive and takes time. Instead, you can have 12 pros sit down, and pretty much 8 out of the 12 will tell you that it's this problem here. You probably know. If it walks like a duck and talks like a duck, it's probably a duck, right? So by doing that, we can save a lot of time and a lot of money. And most often, the judges are usually spot on with their analysis. So that's the second panel we use. The third panel is our difference testing panel. And so that consists of about 50 people in the Institute, almost half of the staff. And maybe once a month we might have a difference test. Now this is an application, like I mentioned before, if you've got an agrochemical product and you want to use it, people are trialing this product on a bunch of grapes. We make wine into it. And then we would do a triangle tests, 2 samples of one of another, try to tell the difference. Based on how many people get that right, above chance, because a third of the time you're going to get it right anyway, we can tell if there's a difference or not. So in a situation like that, that would be a time when we would use a test like that. So that would be our third panel. And then actually, I guess we have four because we also have a consumer panel that we use, which is just a database of consumers that we draw from when we have a study that requires a consumer element. So that's our sensor group. That keeps us pretty busy. Last year we did, I think we had 24 different descriptive analysis studies. And that was certainly the most that we've ever done before. And each year that number seems to keep going up. The demand seems to be increasing and increasing, which is good to see. I think sensory is a component of, to me, and I think to probably Gary, it seems obvious that sensory would be a pretty big thing in wine, yeah? Like, it doesn't seem too much of a stretch. But in industry, it seems that there's kind of the uptake hasn't been the same as it has been in other industries. If you look at the food industry, it is just all product development, quality control, all that stuff is super rigorously tested through sensory. That doesn't seem to happen in wine. There is and I'll sort of be the first to admit, there is an element of personal assessment when it comes to wine quality, and it is difficult to measure. When you're sitting there having a really nice bottle of wine with your friends and a good meal, that is a very large influence on how much you might like or dislike a bottle of wine. So measuring that can be difficult. Certainly, I understand that, but through industry, there hasn't been that same kind of uptake that there has been in some other industries. So it's good to see that these things are people are starting to see the value in sensory, and we're starting to see more and more work come through. So promising. That's good because I'm going to keep my job probably. So I'll just take you through a little bit of research that we did we don't do a whole lot of sensory research ourselves because we are, to a certain extent, a service provider in the Institute. We're quite busy providing sensory for most of the other projects. It doesn't leave a lot of time for us to do our own research, but from time to time we kind of throw out some projects. And so this is one that we did last year on green flavors in Cabernet Sauvignon. So the thing about green is that it's a little bit ambiguous. Like, you know, what do you mean when you say green? When I say banana, you know what I mean. When I say sour, you know what I mean. These are concepts that are well understood, right? They're well grasped. Same with green color. I don't think anyone's going to dispute. We understand what we mean when we're talking about those things. But it isn't exactly the same with green flavor. What do we mean? Green's a color. How can it be a flavor? But this is something that is kind of instilled in the lexicon in certainly winemakers in Australia, and I can only speak to the winemakers there. I don't know if it would be here, but I would assume that it gets batted around a little bit. So we know that there's already some green flavors that exist in Cabernet Sauvignon. We've got our methoxypyrazine groups. We've got cineole, something that we see a lot in wines in Australia. It's that eucalyptus smell. DMS. Hexanol and other C6 compounds certainly give us plenty of that fresh cut grass compound. And we also see characters like herbs or leafy or stalky. These are the things that we see in that you might label as a green compound. It might be in that kind of category. We also see palate terms such as green tannin or STEMI. I think we've all heard that before. What does green tannin mean? So, or underripe is certainly something that's been associated with that, high acid wines. So, how are we going to measure it? What we decided that we would do is we would use 3 different panels looking at the concept in 3 different ways, and then try to marry them all up together and see what they spit out. And then finally, looking at kind of once we'd kind of covered the basis from a sensory aspect, we'd look at the chemistry behind the wines and then try to marry up the sensory and the chemistry. So starting with our descriptive analysis, we took about 40 wines, looked at all of them, picked out 18 that we thought, or not quite 18, I think it was about 16 that we picked out that were clearly green or had different green characters from a group of in-house, using our quality panel, checking their notes and looking at how many people agreed that these particular wines are green. 8 regions in Australia, from $13 to $45. We didn't particularly choose any wines that were supposed to be typical of the region that they came from. So we didn't say, okay, we need the X amount of wines from this region, X amount of wines. We just picked out the green ones, and we got our 8 regions. So we had 9 panelists, we had 8 sessions. Normally, this 8 sessions would be probably a normal size descriptive analysis. We'd have 4 training sessions. The first session, we kind of go through all of the wines. The panelists would write down notes, so we're generating attributes. The second, I would compile all of those attributes, make a big list, aroma and palate. The second day, we would try to refine that list. We would look at a different set of wines, see, does that list that we've created, does it fit? Do we have the right words to describe these wines? Do we need to add some? Do we need to take some away? So that's what we do our second day. Our third day, again, is doing the same, refining the panel, refining the attribute list. And then the fourth day, we go into the booths and we practice with the set that we have. So it's kind of like a dress rehearsal, if you will, with the wines. And then we look at the data and see if, in fact, our list is working. Do we need to make any adjustments? Do we need to change anything? So those are training sessions. Once they're done, then we go into the booths and we assess the wines formally. We did this over 4 sessions. And here you can see a list of the green terms that we ended up keeping. Of course, we would have had fruit terms, oak terms. We had, palate palate terms, acidity, stringency, viscosity, all of these terms that you would use to describe a wine normally. But then we had also these green terms that we added in. And here's a list of our appearance and aroma characters. So what's important about this is, of course, that we have the name of the term. But what's really important when it comes to descriptive analysis is, what have you defined the term as? So green stalks, we would make the assumption that it includes green stalks. But actually, it also includes leaves and green capsicum. So this is the kind of important things to know. So when the panelist sees green capsicum in a wine, this is the scale that he or she is going to rate them in. You could call the term whatever you wanted to. It doesn't really matter as much what you call it. It's important how you define the term. That's what's important to the panelists, because that's where they're going to rate those attributes when they see them. So you can have a look at those. We're covering a pretty broad spectrum of attributes. And so this is the results. So we use principal component analysis to kind of tell us a bit of a story between the attributes, or to tie the attributes and the wines that we selected together. So when you look at these, there's a couple of different things to keep in mind. First, when it comes to the attributes, maybe just a quick refresher on principal component analysis. We've got a couple axes here. And these axes, if you will, it's like almost like a three-dimensional space. There's multiple axes moving all over the place. And these are dots in those three-dimensional space. Can't look at it in 3 dimensions, of course, because we put it on that. But when we show 2 of these dimensions, we can see the vectors kind of pointing out in the into the space, depending on, well, we've got principal component 3 up here, and we've got 1 here. I didn't show 2 because it's less interesting. But what's cool about this picture, principal component 1, this axis here, has separated the wines into kind of 2 little groups, or 2 little groups that I like to point out anyway. We've got this little group here, the 2 Langhorne Creek wines 1 and 2, Yarra Valley 1, Coonawarra 1, and Margaret River 1. These wines here seem to be associating with these attributes here: green stalks on the palate, vegetal, bitter. And we've got this group over here, Coonawarra, Clare, and Adelaide Hills. These ones are being more associated with overall fruit, dark fruit, vanilla, chocolate, color, intensity. One thing about principal component analysis is when you've got these attributes here, these attributes would be negatively correlated with these attributes. So meaning, if these wines here are rated high for these attributes, they would have been rated low in these attributes. So here we go. We've got our riper, darker fruit, bigger styles, and here we've got vegetal, green stalks, and negatively correlated for overall fruit, dark fruit. Up here, in the other direction, we've got eucalypts and mint and herbs. So we've got this one little green kind of quadrant here, and this other one that's up here that really isn't related to these at all. So we've got 2 little groups of green. So that's basically what the principle, or the descriptive analysis has given us.
I'm just going to jump in and
yep.
just ask a few questions to the end, just in the interests of time, please.
Okay. The next panel that we used is a winemaking panel. So we took this concept, or this technique called projective mapping, or napping. And this is a technique that's reasonably new. The French word for tablecloth is nappe. And if you think about a tablecloth, the kind of classic European tablecloth that's red and white checks, seen it in an Italian restaurant before. The technique started with people tasting, or tasting anything, it didn't start with wine, but tasting things and placing them on the tablecloth, and by measuring their locations on the tablecloth through the checks, they'd get coordinates. And by taking those coordinates, you can make a map. And so this technique has advanced a little bit. But what this technique is really good for is that it doesn't require any training, and it's quite quick. We call it quick and dirty, because you don't get as robust results as you get something with descriptive analysis. But this can be done in a day, where with descriptive analysis that took us more than 2 weeks to complete. So we use this technique with 20 winemakers. We spent a day out in McLaren Vale, and we invited a whole bunch of winemakers to come in and do this tasting with us. So we used the same 18 wines, same 18 Cabernets that we used in descriptive analysis, and we had them all taste and smell each wine and place them on a 60 by 90 sheet of paper. They would write the number, a 3-digit code. You can see a 3-digit code on all the glasses here. They would write the number where they'd placed the wine, and then we'd have them write some comments beside the wines on why they had put them there. So we had them do that twice, so we ended up with 40 maps. Now, the thing about winemakers, I don't know how much interaction you've had with them, but they certainly are very verbose when it comes to describing wines. They came up with 120 different words for describing the wines. So that was ridiculous. So I said, all right, the word had to have been used 4 times. That was our criteria for it making the cut. And that still left us with 52, I believe. So that's kind of what they would say is a dog's breakfast. But there's a couple of things that are pretty cool about this. Do you remember those, when we looked at the PCA of the descriptive analysis, we had 2 little groups. Well, here are 2 little groups. Again, these are the same wines, and they're separated the same way. And let's look at the characters that we see in here. Green tannin, earthy, simple, green. And over on this side, we have dense, oak, tannin, full body. So we're starting to see a trend here. The winemakers have separated the wines the same way that the panelists, sensory panelists, have separated the wines, only that they're using some different words. So then we went to our consumers. We took 113. We chose 6 wines using segmentation analysis. So we took our original descriptive analysis data, and we, using segmentation analysis, we split them up into groups of 3, and then we chose a wine from each one of those little groups. So that should, in theory, give us a wine, give us one wine that is kind of appropriate for the group that it was in. And we got our consumers in. We asked them how they liked the wine, and then we asked them how likely they would be to buy it, and collected demographic data and all those kind of things that you would normally do when you meet with consumers. We had to come into to the booths at the AWRI. We don't do this at home or at your wine store or anything like that. So we do screen the consumers to see that they are not, in fact, wine professionals. You would be amazed to see how many winemakers actually show up to be on consumer panels. It is amazing. Probably half of the people we have to, it's amazing, well, because people go to the AWRI, usually you're in some aspect into wine if you go to the AWRI website. So they just kind of click, oh yeah, I'll sign up. So we have to reject so many people. No, you can't be on our panel. You know far too much. That's the problem with winemakers, right? They know everything, so you can't have them on panels because they are far too biased. So our consumers, they segmented into 4 different clusters. Now, this is not surprising, and you would expect them to segment into some clusters. The largest one really liked red fruit and overall fruit. The second ones were more kind of traditional Cabernet likers. The third ones were all about dark fruit. And the fourth one was just a small cluster. It was kind of bizarre. This is what's interesting about this. And like I said, here we have our clusters. I'm less interested in the clusters. What I'm interested in, this vector here, this is the population. So this is the overall liking of the population for all of the wines together. Now, we see some of the attributes that are kind of linked with this. We see our fruits here, and this was the most liked wine, by the way, and this one was second. But what's really interesting here from this stuff isn't what's associated here, it's what's negatively associated with this here. We've got green stalks acid, green stalks aroma, mint, eucalypt, and vegetal. So consumers don't like that. They're not enjoying those attributes. So that was kind of, to me, that was the most interesting part of the consumer exercise. So putting it all together, our DA gave us our two groups of attributes. Our projected mapping showed us that when winemakers are saying the terms green and green tannin, they're generally talking about green stalks, aroma, and on the palate, vegetal and bitter characters. And our consumer panel, they don't like any of them. They do not like green flavors in their wines. And so we just actually repeated this with Shiraz this year. What's interesting about that is that I think that we probably understand green in Cabernet a little bit better because we know about compounds like methoxypyrazines, cineol, and EMS, these characters. We know where they're well characterized, we know them, we've seen them lots. Shiraz is a little bit different. There isn't methoxypyrazines in Shiraz, or normally there isn't. Some of these characters aren't as well defined. And so, and certainly there isn't, when it comes to varietal character, I don't think there's a lot of characters that we would call green in Shiraz. So this has been quite interesting. So we're just looking at the data from this right now. So Treasury Wine Estates helped out with this project, giving us some wine and some winemakers. Wine Australia, of course, they pay the bills. We have to thank them when anytime that we're looking at this research. But yeah, that's what I've got for you today. Anyone have any questions?
I'm going to pass, I think it was going to pass the microphone.
Sure.
If we could ask the questions for our outside audience with the mic, please.
Thank you. I had two questions. Okay. The first one was about the PCA from the descriptive analysis. Mm-hmm. Does that show aroma attributes or taste attributes or both?
Both. So you can hear this. There's green stalks palate.
Oh, okay.
And there's green stalks aroma.
Right. And did you use the same attributes for aroma and palate?
No, no. They come up with their own. The panel comes up with their own. Mm-hmm. Now, certainly when it comes to aroma characteristics, that list is normally longer mm-hmm. than what's on the palate. And often they might have, in the training, we might have green stalks aroma and a green stalks palate. We might find that they're telling us the same thing, mm-hmm. And therefore we only need one. You know, we might, so I was saying we do a practice booth session where we do some training in the booths. We might go into the booths and see, collect all the data from a training session. Then we would look at and do a PCA, and then we would look at correlations between the two from attribute, the mm-hmm. aroma attributes and the palate attributes. Oh, okay. And so if both vectors are pointing in exactly the same spot, mm-hmm. really, we don't need to, because the more attributes they have, the more harder it is for your descriptive analysis panelists mm-hmm. to separate. It's one more thing for them to worry about. So we usually try to streamline that pretty good. If they're telling us the same thing, we don't really need them.
Yeah. Thank you. And I have one more question, please. Thank you. Short one for the training, for the descriptive analysis. You mentioned 4 sessions. Do you find that's enough in your experience?
Yep. Yeah, I do.
Do you train
Now,
on?
again, these are, our panelists are professionals, right? So they do this.
Oh, okay. That makes sense.
They do this every, all year.
Thank you.
Every year.
Thank you.
You don't want to hand the mic back to me. I've got a whole page of questions, so please, for Wes's sake, other questions?
Please.
Hi. I saw on your consumer preference that you did a PCA.
Yep.
And I was just wondering, did you supply the consumers with a list of attributes, or did they come up with those on their own?
No, so those attributes, those are attributes straight from the descriptive analysis PCA.
Okay, so that you could have some...
So we've done partial least squares analysis and taken the attributes from descriptive analysis and tied the two sets of data together. So we've tied the consumer data into the descriptive analysis.
So it's kind of like an overlaid PCA?
Yeah, that's right.
Okay, thank you so much.
Cool. Thanks for the talk, Wes. I learned a lot, and it's some really interesting stuff. I've got several questions, but my first question, playing the devil's advocate, I always get concerned when I see such general rejection of a particular group of flavours, and this last PCA that you finished on, and your interpretation too, is essentially that there's these 4 clusters, or perhaps more in the data, and all of them are rejecting green flavoured wines. Or we could be more positive in saying they're accepting, ripe fruit flavoured wines, but in the context of how you're positioning this, they're rejecting green flavoured wines. So a couple of things. First of all, to what extent is that behaviour related to the wine experience or involvement of consumers? So one could argue that the more familiar and experienced or expert we are with wine, including Cabernet-based wines, the more likely we are, in fact, to reject that, because Cabernet from Australia surely, should never be green. So I wondered to what extent consumer experience and expertise might factor into that. And maybe you've got that data. And secondly, again, playing the devil's advocate, these are South Australian wine drinkers. Would you expect the same results in a different region or even different country? So actually it's a bad example, but let's say you did this in Tasmania. There's not much Cabernet grown in Tasmania, but would you expect that same rejection of green flavours? So A, as a function of one's involvement and expertise and knowledge, and secondly, would these results hold up, you think, in other regions?
Let's see. First answer, I would probably, or answer to your first question, I would say that, in my opinion, I think wine experience would play a great role in the rejection of these flavors. But I would think it would come from the other end. I think they would be less, they would reject these wines less, these attributes less with experience. It's been certainly it's been my experience that consumers in Australia, from a very kind of base level, they don't like astringency, and I think that one, yeah, definitely against they generally don't like astringency. They like fuller-bodied, richer styles, even sweet wines, and so it stands to reason that these are negatively correlated with their liking. However, as you get more experienced and understand structure in wines and tannin, I think appreciation of certainly a variety like Cabernet and its varietal character, and those green characters that go along with it, I would expect acceptance to increase. However, our consumer panel, it's meant to be reasonably general in its scope. I think 116 people that we had through. And like I said, we do screen them on the AWI website for no wine experience, no professional wine experience, and just very basic tasting parameters.
So wine experience or self-rated expertise wasn't data that was collected from these people to say, test that idea out?
No.
Okay.
No, we didn't ask them if they thought they were, what level of expertise they thought they had. Part 2? Sorry, refresh my memory on part 2.
Sure, I was just wondering how generalisable do you think these results are?
Of course, region.
Consumer groups, some different, yeah.
Hard to know. And as you clearly pointed out, these are South Australian consumers. We live in a warm region. Generally, consumers tend to drink what is produced around them, right? Normally, these consumers would be consuming South Australian wines. If you are an average consumer, you're not buying Yarra Pinot if you live in South Australia. So I would think that their preferences would lie in that style I mentioned before, riper, richer, less tannin, more fruit-driven. If you live in Tassie, well, that would be Tasmania. You may be more open or used. You might see it more in kind of everyday wines that you drink. If you drink local wine, you may see wines that are more acid-driven, less ripe, more tannic, stringent, etc. So, quite frankly, I'd be surprised if there wasn't sort of an effect, but we haven't tested that.
Thank you. Just a follow up until I see other hands being raised, and with. I think one of the interesting things about this sort of work is that nexus between sensory winemaking and consumer. And again, this graph is a great starting point for that. Marketers, I think, would be particularly interested in your red text in these 4 clusters, and in what, if any, characteristic demographic features or other factors might constitute those clusters. So I'm wondering, so for instance, perhaps cluster 1 was primarily 18 to 24-year-old females, as an example, and therefore that's a marketable, actionable consumer segment in terms of both winemaking, so fine-tuning the production or the blend, but also the branding and advertising and promotional activities. So I guess my question is, have you or will you look at those clusters in terms of characteristic demographics or other factors?
We looked at them pretty briefly. Now, when it came to this study is multimodal. There's a fair bit going on, and there certainly wasn't a lot of depth when it came to the consumer, questions. I certainly see the benefits in having a really nice kind of demographic, or a box that we can put from this consumer likes this and likes this. It's been my experience that that's hard to do. A lot of these kind of consumer groups are pretty dynamic and move really quite quickly through these different clusters. That's from the work that I've seen from people like the larger wine companies that have the work that they've done. It's harder to put your finger on who actually fits in these clusters. But for what we did, we didn't really look too much into what was driving the cluster from a demographics point of view. From age, sex, things like that, there wasn't anything that moved through or that stood out. But we didn't. But from the data collection point of view, we didn't really collect that much information.
Thank you.
I guess my question just sort of builds on what we've been talking about. I was really fascinated how cluster 2 separates out, I think it's on principal component 1, from 4, and is it 4 and 1? Yeah, 4 and 1 and 2 seem to be significantly different, and that principal component accounts for a lot of the variation. And I'm wondering if you could comment possibly on the nature of the green characters that make up, so possibly, green stalks and vegetal might speak more to the population in general rejecting it, whereas the things that are more closely tied to principal component 1 might separate the 2 cluster groups. Like, do you think that all green character can be considered in general, or do you think they kind of group out and maybe we're looking at, there's a population trend, but then within there's certain characteristics that talk about group differences or cluster differences?
Well, I think like with cluster 2, I think we called the cluster 2 the Cabernet likers. And they like this eucalyptus and mint. They still like that robust, rich, dark fruit and opacity, but they just didn't seem that they didn't seem to be as adverse to some of the stocky characters that were in there. Yeah, these clusters and what people like and how they like it, I think they're so dynamic, and it's really hard to put your finger on exactly what it is that that's driving them. But clearly, there is a classic Cabernet element to this group with what they like when you look at the attributes that they kind of end up being associated with, for sure.
That's really interesting. It's just amazing how clearly different some of these clusters are on that component, and how much of the variation, at least in this data set, it accounts for.
Sure. Certainly with that one anyway, there was lots, like if you looked at, and this was something that we ran into, we ran into with the study. There was, looking at that data, it told us that it was reasonably robust. Mm-hmm. We were getting somewhere with the data. You look at the winemakers, and this is kind of what I was mentioning before with winemakers. They just you don't know what. It's very difficult to pin down what they're saying. There's really not a lot being explained here. There's still a lot of ambiguity out there. And when we looked at every winemaker's map versus the average map, the R-squared values were just like there isn't a lot of winemakers that are saying the same thing.
Hi, Wes.
Hi.
One of the things, though, with the winemakers is...
So they haven't been, I think from my understanding, they haven't been directed to split it between aroma and palate. So from the napping, it looks to me as if they can use anything they want, aromas, flavours. So that's all in together, and I just wondered how different that would be if they were allowed to focus on, say, just flavour, so more partial napping, and whether that would see a bit more
We've done that before with when we've kind of been testing this methodology out. Do we just do aroma only or just flavour? Do we collect these terms? Adding the terms onto the map is something that we've only done for a couple of years, simply because the data, the software package that we use to analyze this data is incredibly complex and difficult dealing with the data. Two different, when you're talking about, these aren't coordinates, so to speak, right? They're words. And so you're tying in with a word with a coordinate. It becomes extremely complex. So we thought long and hard about saying, look, let's just do this by aroma, by palate. But the descriptive analysis was done with both at the end of the day. And so that was why we decided, look, let's just give them free choice. They can describe the wines how they want.
Okay.
So, but certainly, like from what we've done in the past, it's way easier if you say to your panel, if you're just going to use this this technique or this process in situ, it works well, because you're making it less complicated for your judges, and from your analysis perspective as well.
A couple of actually, I see Andrea's hands up, so I'll give it to her.
Yeah. Were there any chemical analyses done to these wines? Did you look?
Yeah. Oh, there's been a huge suite of chemical analysis that were done. Your basic chemistry has all been done. But once we got through all of this stuff, then we started looking at, in particular, these wines. Mm-hmm. We started collecting tannin data on those wines, and seeing what did they have in common. What were they high in, what were they low in, etc. Stuff also like in measuring in aroma compounds. These two wines, for instance, they were highest in cineol, which is the compound responsible for eucalyptus. So we've collected all that data, and it's all getting put together, and we're writing it all up. But we want to get the sensory down first. And then it was kind of like I was saying before, like we did with our expert tasting panel. You get the sensory done first, and then you know where to look.
No, I thought it would have been interesting to see something like this, a PCA with the descriptors and the chemical overlap. Yeah.
Thank you.
I just want to follow up on Belinda's question related to methodology. And I guess the first question is, with the projection mapping instructions to the panel, are they asked to group the wines according to similarity or dissimilarity? So, or what exactly are they asked to focus on in terms of making their decisions as to where to group the wines?
Both. The instructions are that to taste, you taste your first wine or taste a wine, place it on the map, and you take the next wine and you place it as close to or away from, depending on how similar or dissimilar it is. So it's not so you don't say it's not one or the other. It's in fact you want to. I do explain to them it's all about how far or close things are together. So it's about the distances between the wines. That's what it's about. Where you put them is less interesting. It's how they all fit together from being close to each other or apart from each other.
So that's an interesting, because I was intrigued, and I think it was part of what you were concluding too, how similar the DA results, at least in terms of this graph, mirror the projection mapping results. And I was wondering cognitively how similar the methods are, because DA is all about, including the statistics, is all about maximising differences between samples. And from a perceptual perspective, people tend to apply different cognitive processes when they're asked to rate the similarity or to group things according to similarity compared to differences. There's actually different processes. So it's intriguing to me just from a basic perspective that these maps or solutions are so similar.
When we give them instructions, I tell them that you're creating a map. And that's kind of a concept that I want them to understand when they think about it, is that you're making a map. And it's not, it's less a grouping or sorting task. That's not, grouping those words I don't mention them, because I don't want them to think that they have to put wines together in a group or anything like that. It's just placing the wines on the map based on how similar or dissimilar they are with with their next neighbor.
Can I just add on to that? Is that
Thank you. One of the things I found when we did partial napping here with ultra flash profiling, so they use their own terms, was that those people who are familiar with a PCA actually draw a PCA on it, and they're actually thinking of a PCA when they're doing it. And so we had to eliminate that in the first place, just because they have that picture straight away, and they know it's going to be transformed into a PCA.
That's really
and they're kind of being asked to put a PCA in front of them. So there was a element of bias I found with the napping when we started, or in the results, that I couldn't use for that simple reason. And I just wonder whether you've ever come across something like that people who know of it.
I haven't, but again, we use our descriptive analysis panel. That's kind of who they've been the guinea pigs for trying this stuff. So they don't understand statistical analysis. They don't have a basis in that, or wine tasting, or anything. They just have, in fact, it was tough to get them to do this because they do DA so often. Yeah. They get regimented into the format that they're using, and then what is this? And so at first we had them do it on the paper, and we use Fizz sensory software for data collection, and we'd have them do their map, and then we would have them transcribe that onto the screen. Now, what we didn't realize is that some people are good at that and some people are not good at that. And so now their map that they put up on their screen doesn't actually represent the map that they've got here. So that was a problem that we ran into. So we eliminated the map and just started doing it straight on the screen. But like I said, we don't have and winemakers don't really understand PCA either. So They may, but it's when we presented this in front of them, it wasn't something that it wasn't anything we could see and go, oh, look, that guy's got a clue. He knows exactly what's going on here. That wasn't it wasn't the case at all. But that's really interesting to hear that.
We've got time for 2 more quick questions, assuming that there's nothing from our online audience. Margaret and Gary to finish.
I guess just picking up on what Belinda was talking about, is there an effect when you use this technique about the wine that you start with? So if you're always sort of comparing wine 1 to wine 2 and then to wine 3, do you have to randomize the order that people are tasting the wines? Or, because if I was a naive consumer, I've never done this technique, I would probably take the first wine, plop it right in the middle. Now I'm fairly familiar with PCA, which is probably speaking to that, but I'd plop it right in the middle and then start working my way. So if I started with wine A as opposed to B, my map would look possibly completely different.
Absolutely, for sure. And so we randomize the wines when they go into the booths, right? So that wine, in position 1 or the first closest to them and on the left is different. So all the wines on the tray are different. But really after that, as long as you can get them to all start with something different, after that I think randomization is moot. I don't think it matters that much. But that first one, absolutely. And so we used to randomize everything, and we used to tell people you need to go through and organize them in this way or whatever. But when you're in a sensory booth, and probably most of us have been in a sensory booth, it's not that big. So when you've got, and this is a methodology where we like to use, we might do between, say, 10 and I think 18 is the most that we've ever used. It's getting up there. But there's not a lot of room. So having them go through exactly the same way every time, when really, like I said, really getting them to start with a different one is the only really one I think is that important. So that's kind of what we focus on. We say after that, go grab them however you want.
Very patient with us, Wes. I'm going to sneak in a very quick last question. and that's this. Do you have a sense of how variable either this panel or in general consumers are in terms of their greenness perception? And I asked that, our lab's very interested in greenness and in differences between individuals. And so, for instance, pyrazine sensitivity, there can be a 300-fold difference between one consumer and the next in terms of their sensitivity to these green compounds. Do you have a sense for, and of course one might predict that would influence the segmentation and perhaps also the rejection of green flavours so do you have a sense of if that's operational in general for Australian consumers or for your panel in particular?
Well, it would be naive to think that it's not. The level at which that we're measuring, these things, and certainly the kind of depth that we have with our consumer panel and our questions and things like that aren't covering things anywhere near that kind of level. I think that would be a really interesting direction to take it. Certainly based on some of the work that you've done, and there's huge variance between the consumers that is basically kind of it's not the elephant in the room, but do you know what I mean? We don't really it's not given enough credit that there's huge genetic variance in what consumer A versus consumer B can smell, taste, and their perception and liking based on these kind of things. So I think this would be huge influences. But no, it hasn't been something that we've looked at this far.
Well, Wes, we need to wrap things up. We could go on for hours. I think we will go on for hours afterwards, but in terms of our podcast commitments, we need to wrap things up. It's been a fascinating talk, evidenced in part by how many questions there have been and there still are. Thank you very much, and here's a small token of our appreciation for travelling here and for your talk.
Thanks very much, Gary.